are in the thalamus. So we've got this green neuron, the secondary sensory neuron. Its cell body and dendrites are in the lateral horn, or the dorsal horn. It's crossing over to the white matter, going up the white matter to the thalamus, and that's where the synaptic terminals are. Then there's a third neuron that starts there at the thalamus and goes up to the cerebrum. So we'll call this the third order sensory neuron. It's also multipolar. So the cell body and the dendrites are in the thalamus.
For the third sensory neuron?
Yes. Our third order neuron, the cell body and dendrites are in the thalamus. And the synaptic terminals are, where does this piece of information get the conscious awareness? What part of the brain receives sensory information?
Cerebrum.
It's the cerebrum, more specifically.
Somatic sensory cortex.
The primary somatosensory cortex, where's that located?
In the parietal.
In the parietal lobe. Remember, right behind the central sulcus? So that's where this is ending. So it's ending at it says cerebral cortex. Specifically, we mean the sensory homunculus, the little person. Remember I showed you that little person? Homunculus. And the primary somatosensory cortex. Homunculus.
Is that homo or
H-O-M-U.
U? Okay.
So the cell bodies and the dendrites were in the thalamus. The axon was in a tract, and the synaptic terminals will be in the primary somatosensory cortex. So this is our third order sensory neuron. You know how we talked about the thalamus? Its job as a relay and filtering station? Like a good secretary that keeps a lot of information from getting to your conscious awareness, like the fact that you still have clothes pressing on your skin. Now we can really see the anatomy behind that idea, that you've got these mechanoreceptors in your skin that are being stimulated by the weight of your clothes on your skin. And that information is going across here, and neurotransmitters are being released, and we're getting an action potential here, and that's going up, and it's getting to the thalamus, and the thalamus is saying, okay, this is really not worth bothering the cerebrum with. And so the pathway kind of ends there in the thalamus until there's a reason for you to have to pay attention to it. Yes?
The synaptic terminal on top, is that the cerebral cortex?
Yes, in the primary somatosensory cortex, which is part of the cerebral cortex.
Okay.
one major sensory route information to the brain. There's another one, another way where fine touch, so this is called the posterior column pathway. Fine touch and proprioception goes that way. Remember, proprioception is the information about where your hand is, your body is in space. So if you're reading Braille, that's going to require sensitive discernment. That's going to go up this way. Or if you're closing your eyes and knowing that your hand is above your head, that's going to go up through this pathway. So this is basically the same kind of idea. The biggest difference is that first order neuron doesn't synapse in the spinal cord. Instead, it just goes up the white matter. It ascends, meaning it goes up, and then there's this vocabulary word, ipsilaterally, that's an I. This is one of your vocab words in your packet. Ipsilaterally means on the same side. The opposite of ipsilaterally is contralaterally, meaning on the opposite side. So this means it hasn't crossed over yet. It's going into the gray matter, but then it's coming out into the white matter. It doesn't synapse here. It stays on this side, goes up to the medulla oblongata. And that's where the synaptic terminals would be. And then the second order sensory neuron starts there. So the second order sensory neuron crosses to the contralateral side. And then from here, it's basically the same because it goes up to the thalamus, where the third order neuron goes to the cerebrum. So if we kind of compare them, how many neurons does each take? 3. It takes 3 neurons. Where do they cross over? In the spinothalamic, the crossover Or where's the first synapse? Okay, so we could say number where the synapse is. Spinothalamic, the first one synapsed in the dorsal horn, and then in the thalamus. And the posterior column synapsed in the medulla, and then the thalamus. In both cases, it was the second order sensory neuron that did the crossing over. It's just that one crossed over in the spinal cord, because that's what the synapse was, and one crossed over in the medulla. Okay, so then how the brain interprets this, the interpretation of the stimulus? The brain has to figure out where in the body is the stimulus coming from. And we already talked about how it knows. How does it know that this stimulus is coming from the big toe? Yeah, but how does the brain know that? The homunculus. Remember how there's that map on the cerebrum? If it's coming from the big toe, the brain knows it's coming from the big toe because it goes to the big toe area of the sensory homunculus in the cerebrum. So it's where in the sensory homunculus the action potentials arrive. How does the brain know if this is a moderate stimulus or a more intense stimulus? Does this hurt a little bit or does it hurt a whole lot? Larger stimulus equals larger action potentials. No, it doesn't. All action potentials are the same size. Action potentials are all or nothing. You either have an action potential or you don't. There's no extra information to be had in an action potential. So it can't be a larger action potential. So can you then guess? If it's not a bigger action potential, how is the brain going to know more? Larger stimulus triggers more action potentials. All action potentials are the same size, so it couldn't be a larger one. But it would be a really reasonable true-false question on the next exam. True-false, explain, right? A larger stimulus, true or false, a larger stimulus triggers a larger action potential. Okay, and then the last thing is the type of stimulus. How does the brain know whether this is temperature or pain? Pressure or temp, like, or temperature or proprioception. Like, how does it know which stimulus it is? It's the labeled line. The action potential arrives on. So your brain basically knows that a particular arriving route, a particular pathway is for particular information. So if it's coming in on a route that's pain information, the brain says, oh, it's pain. If it's coming in on a pressure line, the brain says it's pressure. Coming in on a temperature line, the brain says it's temperature. You can show the limits of this by rubbing your eyes, and you get those flashes of light or color that you get when you rub your eyes. What you're doing is you're stimulating your eyes and sending action potentials down your optic nerve. But they're mechanically generated. You rubbed your eye. But it's coming in on a labeled line that the brain interprets as being vision. Like, it assumes that it's coming in on the vision labeled line. This should be visual information. So the flashes are your brain's trying to make sense of these action potentials and saying, oh, that one there, this must be flashes of light. Like, that's what the brain, the error the brain is making, because it's assuming that anything coming in on a visual line is visual information. Okay, it's well worth your while to practice these pathways. We'll do some practicing in class in a little while. Not today, but just like you really need to know the parts of the sensory or motor neuron, now you really need to know how they link together to get that information up to the brain. So that was the route for general senses, for touch kinds of senses. The other senses are what we call special senses, because they have to have special ways of detecting them. So smell, taste, hearing, vision, balance, those are the special senses. Okay, so in this picture, this is the nasal cavity down here, the inside of your nose. And you've got these little particles that are coming in on the air. So this is what we would call an odorant molecule. It's a little tiny piece of the chocolate chip cookie. It's floating around in the air. So the first thing that happens is the odorant molecule binds to receptors on the cell membrane. Of olfactory neurons. You can see the olfactory neurons, right? You can see them embedded there in the nasal epithelium. the really interesting thing about this, there's quite a story about about the discovery of these. These are proteins. They're binding to, which means they have to be coded for by genes. And there are hundreds of different ones. You can smell hundreds of different odors. So you have about 25,000 genes, and several hundred of them specifically code for making olfactory receptor proteins to allow you to smell all those different things. That's one of the ways they figured out what they were. They figured there must be we knew we could smell a bunch of different things, so they figured there must be a whole lot of receptor genes, and so they went looking for clusters of genes that had some similar structures, and that's how they found them. But when they found them, they discovered that in humans, many of the genes for the receptors have mutations. They don't make functional receptors. In other words, sure, you can smell hundreds of different things, but there's more hundreds of different things that you can't smell because the receptor that you once upon a time had for it has accumulated a mutation and doesn't work anymore. Dogs, on the other hand, or wolves, have hundreds of receptor proteins and they basically all work. And this is what we call relaxed selection. Like if there's a molecule in a chocolate chip cookie that you can't smell, how likely is this going to reduce your ability to survive and reproduce and have surviving offspring if you can't smell all the same number of chocolate chip cookie molecules that a dog could? It's probably not, right? Smell is important to us, but it's not critical to our survival. So if you happen to have a mutation in an olfactory receptor gene, and mutations are random, they just kind of occur, the odds are it's not going to impact your ability to survive and reproduce, which means it gets passed on, and they just kind of accumulate because there's no survival disadvantage to having them. Whereas if you're a predator that hunts by your nose, and you have a broken receptor protein, your odds of surviving and reproducing are less, and therefore natural selection removes those wolves that can't smell the deer up ahead are more likely to die and not pass on those genes, those broken genes. So the thought is that this is due to relaxed selection. As we became more visual, then mutations in smell genes had little effect on survival. Okay, so we've got these odorant molecules and they're binding to receptors on the olfactory neurons. Those are here. Is there clarification? Go ahead.
Is that related or relaxed?
Relaxed. Meaning, natural selection isn't working very much anymore on our ability to smell. And therefore, mutations tend to accumulate. There's no cleaning up by the individuals that have those mutations dying. Instead, they just survive and reproduce and have kids that have mutations. And pretty soon, there's lots of mutations kicking around in the population. Okay. All right. So back to the main point, though. Odorant molecules bind to the receptors in the olfactory neurons. And so here's the olfactory neurons. So they're embedded in the epithelial tissue of the nose. You get to recycle some knowledge here because these receptors are G protein mediated. So that whole sequence of effects, G protein, cyclic AMP, etc., protein kinase. You guys are all really good at this now. What it ends up happening, the final effect, the thing that gets phosphorylated, is that it triggers sodium channels opening, which we know protein kinase phosphorylates things, and that can open channels. That's what's happening. So then we get sodium influx. And if enough sodium comes in that it depolarizes the membrane to threshold, at the initial segment, then we get an action potential. So the action potential travels down the axon. And the axon is in the olfactory cranial nerve. Cranial nerve number 1. Remember, once one takes the anatomy O for olfactory, that's the olfactory cranial nerve.